interrupted mating experiments, right? So as I told you, these conjugation experiments can be used to map the position of genes over bacterial chromosomes, and mostly the HFRF negative conjugations or mating are used to trace the position of genes, right? So this is called interrupted mating experiment because you need about 100 minutes to transfer complete bacterial chromosome along with the plasmid from the donor to the recipient cell. If you hamper or if you stop the transfer in between, you are able to transfer few genes. So what is the basis? So basis is those genes which are present near the origin of transfer, they are having a higher frequency of being transferred from one cell to the another cell. For example, from the donor to the recipient cell. There were two scientists, Wallman and Jacob. They mixed the HFR and F negative strains with suitable markers and assessed the recombination of specific genes at different times. So they have studied this. A culture containing both the types, that's HFR and F negative strains, was incubated, and they were removed at various time intervals, and they were placed in an agitating kitchen blender to break all the conjugation tubes, break all the pilus. Agitation in the blender separate the conjugating bacteria, and therefore the transfer of DNA will be stopped. To assay the cells for genetic recombination following the blender treatment, they have grown them over the some antibiotic media, and because they are having some marker genes, therefore they are able to assess them. So what is the basis behind this? Suppose you are having genes in a sequence A, B, C, D, E, F, G, right? So the genes which are closer to the origin of transfer or IT, for example, A, B, and C, in the order A is having more probability of being transferred because when this plasmid will transfer from 5' to 3' direction, the A will have more probability than B, then C, then D, and then it is followed like this. So the different genes, A can be anything. So the gene which is actually present near ORIT is having more probability or more frequency of being transferred from donor to recipient. The rationale behind the interrupted mating experiment strategy that at the time it is taking for the transfer of genes to the genes to enter a donor cell is directly related to their order, which is actually over the bacterial chromosome. Because this is a circular chromosome, therefore it is always dependent upon this order in which they are present over this bacterial chromosome. And because you are hampering or stopping the transfer of genes at various intervals by using the click in this kitchen blender by agitating the cell cultures. Therefore, suppose you have removed earlier, then A is only being able to transfer, then B, C, and D, and then you will make a library, like you will arrange all the genes so that you are able to move all the cells in this sequence in which they are present. They are actually present over this bacterial chromosome. Yes, if you are having a query, please ask someone. Okay, so in this mapping strategy, it was hypothesized that the chromosome of the donor strain in an HFR meeting is transferred in a linear manner. It is always linear fashion in the order to the recipient strain. The order of genes along the chromosome can be deduced by determining the time it takes various genes to enter in the recipient strain. I will tell you how they have decided the order also. It is given at the end of this discussion. So thus the interruptions of mating, the time factor is very, very important here. The interruption of mating between HFR and F negative at different times would lead to the various length of HFR chromosome. So this is called HFR, in which the F plasmid is integrated with the bacterial chromosome. So at various time intervals, the various lengths of this various length of this bacterial chromosome are able to transfer from donor to the recipient cell, and the different genes along with it in a specific sequence or in order. So if the two bacterial cell had mated for a short period of time, only small segment of HFR chromosome will be able to transfer to the recipient bacterium. But if you allow it for the long period of time before being interrupted, a longer segment of HFR chromosome could be transferred. And if you allow it for 100 minutes or over that, around 100 minutes, for example, it will be able to transfer the complete HFR, and the cell, F negative cell, will ultimately become F positive in that particular situation. Hence, by determining which gene were transferred during the short mating interval and long mating times, the order of gene can be deduced, which are present over here. So, how these two scientists, Wolfman and the second one was actually I forgot on the last page. Jacob, Wallman and Jacob, how they are able to deduce these order of genes over this bacterial chromosome. So suppose here, so before discussing this, actually this is a different topic. So suppose it is having three genes, uh, and you know the F negative cell is having this, all the three genes are absent, and these were sensitive. You need some uh, resistant strains, that is recipient actually. Okay, so there is pilus which is making the cytoplasmic bridge, and these A, B, C, and these are present over this gray portion. These gray portion are bacterial chromosome, and these black portion are F plasmid. And there is an ORIT, origin of transfer, from where these will start transferring. So here, for example, so this is a 5' end of from the origin of transfer, or IT, and in 5 minutes you will see, for example, only gene A is able to transfer from the donor into the recipient. After 10 minutes, A plus B, and then after 15 minutes, you are able to see all the three genes, where A is actually leucine biosynthesis gene, B is lactose fermentation gene, and C is galactose metabolizing or fermentation gene in bacteria. So all are able to transfer within 15 minutes. So therefore, you need to note these time points, which are very, very important during the gene transfer experiments, or in mapping the genes over the bacterial chromosome. Then you will grow them because you have chosen two strains of bacteria. Donor were sensitive, the recipient were resistant, right? So resistant strain will grow over the antibiotic containing media. You will add streptomycin, and you will see only those cells which are resistant, actually the F negative cells, will grow. And then you will check for these leucine, lactose, and galactose biosynthesizing or metabolizing genes by removing one particular component from the media. We have discussed various types of media. If you remove one particular component from the media, then if you are able to grow these cells, they will show that whether these recipient cells which you are receiving are A positive, B positive, or all positive, right? So so if you made them with the wild type counterparts, and recombinants are selected by plating them on the media of streptomycin, you will able to grow those cells which are actually recombinants. Okay. Trishla is continuously having some internet problem. I do not know what kind of. network or which company you are using, but please go to your roof so that. Hey guys, others, are you able to hear me clearly and my screen is visible to you? Please confirm. Yeah, the others are having good network means. Thank you all. Okay, so Trishla, please try to fix it. I cannot do anything. Uh, Either you need to choose some better network. I'm using Geo. It is working very fine here. Okay, so now you are able to understand that time factor is very, very important and how the gene mapping or the order of gene can be deduced by using these interrupted mating experiments from this particular diagram. This seems to be very clear, right? So now we are having one more problem with this HFR experiment, right? So what is the second problem? Second problem is the same bacterial cell may have the same bacterial chromosome may have more than one HFR. What it mean? But it simply mean that the it may have more F plasmids integrated into the bacterial chromosome. So if this type of problem arises, how will you decide that what is the specific order of genes over these chromosome? So let's discuss it in much details. Okay, so the HFR chromosome, originally circular, unwinds and is transferred to the F negative cell in a linear fashion, right? This we know. The transfer begins from the specific point, that is a point at the integrated F plasmid, that is called ORIT, that is origin of transfer. The further a gene is from the ORIT, it will be transferred later. And since different HFR strains have their F plasmid inserted at different sites on the bacterial chromosome, transfer of chromosomal gene begin at different points. So here you can see there may be more than one F plasmids integrated into the same bacterial chromosome. And this kind of multi-HFR condition, you may have some complex situation in which if you were asked to trace the order of genes, then it is quite difficult because you are having more than one uh, F plasmid integrated into the bacterial chromosome, right? So the transfer of chromosomal gene begin at different points. Uh, Suppose from HFR1 it is having ORIT and it will be transferred in this order, or it may have this order A, B, C, D, right? So it is not always that it will happen clockwise or counterclockwise direction. It will always depend upon the orientation. Always the 5' end will be moving first from the donor to recipient cell, and therefore the complementarity will decide whether it will be clockwise or anti-clockwise transfer. For example, see here, these black sites are actually in site of integrations of a plasmid, therefore they will make different HFR. So HFR1, HFR2, HFR3, where so you can say there are three F plasmids which are integrated at various sites in bacterial chromosome, and this is a single bacterial chromosome. Now, HFR1 is actually having A, B, C, D, E, right, in this order. So it is having 4 genes up to D. And okay, so if the origin, if the start is from HFR1, so the order of transfer will be A, B, C, D, right? So it is counterclockwise order. Gene A will be enter first. If you look at HFR2, here you will see that the order is in this direction, F, G, H, I, J, right? So this is also counterclockwise. But if you look at HFR3, you will find that it is transferring in F, G, H, I, J in this way. So this is clockwise order in which the uh, gene J will enter first because it is very near to the origin of transfer. So one thing you need to remember in always, the gene which is close to the origin of transfer will be transferred first, right? So here you may have two possibilities, that either A will be transferred first or M will be transferred first. So always some clue will be given in the question if this type of question has been asked. Okay. So consequently, the gene transfer may be either clockwise or counterclockwise for any particular HFR strain, which are present in more than one copy in a single bacterial chromosome. The following example can illustrate how the three genes leucin, lac, and galactose is mapped over HFR interrupted mating. So you may have the two strains, and then you will note various time points, and then you will grow them over some media which is actually do not having this leucin, lactose, or galactose. And okay, so if it is not having leucin, if it is leucin positive, and to confirm whether the A gene is being transferred, so you will grow the cells over a media which is actually do not having leucin amino acid. If it is leucin positive and the leucin gene is transferred, so this cell is able to synthesize the leucin without being added into the media. If you add the lactose, and if you do not add glucose or some other source, so the bacteria will survive by degrading or metabolizing the lactose. Similarly, if you add galactose, still if the gene C is being transferred, it can metabolize the galactose. Otherwise, the if you add galactose, it will not be able to survive over the galactose medium. So that's how you will grow these cultures, and you will decide which genes are transferred, which genes are not able to transfer, and what are the time points in which these genes can be transferred from the donor cell into the recipient cell. And then you will make this type of graph in which you show that in 5 minutes all the A genes are transferred, because the A gene is very closely linked to the origin of transfer. Then B is actually lactose, C is galactose, and that's how this is the linear fashion in which genes are transferred from one bacterial cell donor to the recipient. There is table of mechanism of DNA transfer, and this is showing some important facts which we have discussed in our 2-3 classes. Transformation. In this, the naked DNA transferred, right? The nucleic acid which are present in the environment are uptaken by the bacterial cell by some proteins which help in the binding of DNA to the bacterial cell surface. Size of DNA transferred, it is a part of chromosome. There is no polarity, and it can transfer in either order, 5' to 3' or 3' to 5'. No, nothing is like polarity there. Sensitivity to DNAs, yes, they are sensitive to DNAs because they are present outside the cell. Therefore, there are chances of degradation. Also, since they are transferred, only the single strand is transferring. Okay, during transformation, only the single strand is transferred to the bacterial cell cytosol. As I told you, there are some exonucleases, they will degrade the second strand. Therefore, because they are single stranded, they are very sensitive, they are very unstable, and they are sensitive to DNAs. In transduction, this is happening with the help of bacteriophages or some viruses. We will discuss M13 and lambda phage. Okay, so the DNA enclosed in a phage capsid, it is having some protection. So they are present in the viral capsid or coat. They are part of chromosome because some of the genes are only uptaken from the bacterial chromosome. There is no polarity. They can be transferred in either orientation. And since they are protected with this capsid, they are not sensitive to the DNAs. Also, they are double-stranded, right? They are stable too. In conjugation, it requires cell-to-cell direct contact, and F plasmid or part of the chromosome can be transferred, and complete F plasmids part of the chromosome along with the F plasmid, that is called HFR, can be transferred. Polarity, yes, it is always only this is having polarity in transfer, that is 5' to 3'. And since it is double stranded and they're inside the cell, therefore it is also not sensitive to DNAs. Now there is a problem in your book also. So there are two problems. In fact, one is Several high-frequency recombinant strains are mated with an auxotropic. What is auxotrop? I told you, auxotropic strains are those. So we have discussed two strains. One is auxotroph, one is prototroph. Auxotrophic, prototrophic. So auxotrophic are those strains which are able to which are not able to synthesize some specific component, and therefore you need to add that particular component in the media, right? Whereas the prototrophic strains are those which are wild type, and they do not having mutations, therefore they can synthesize all the essential amino acids and other factors which are required for survival. Order of transfer is given. So strain 1, threonine, this LIP, tryptophan, histidine, threonine. Strain 2, strain 3, strain 4. So actually, sorry. So what you need to find, the order of transfer of genes. Only two things you need to remember in such kind of problems. One, they will always travel or transfer in a linear fashion, because there is a polarity, right? Second thing you need to remember in such kind of experiments is, okay, so first of all, you will see all the strains and you will try to arrange them in a specific sequence, because these strains will show you that these HFR strains, so they can be in either orientation. As I told you, they can be clockwise or counterclockwise. So you need to make them in a specific sequence. So first of all, if you write threonine, LIP, tryptophan, histidine, and THY, I do not know, this is threonine, I think. So if you do not know, still you do not worry. What you need to do, you will need to arrange them in a specific sequence. Suppose this is the sequence for one strain, you will write them in this order. Then for the second strain, you will match this THR. Do you have THR in the second strain? No. So something common, histidine. You are having histidine, right? And after histidine, if you see this, you are having threonine, THY, but actually you see here you are having threonine on this side here. So what you will find from this second strain, actually you need to move in this order to make this linear, right? So if you see this, so this is order: THR, LIP, tryptophan, histidine, threonine. Here you are having this LIP, right? Tryptophan, histidine, threonine, THY, and STR. Means what it is showing you? You need to write STR here. So you will write STR here, right? Now what will you do? In the next strain, you will try to find the sequence. It can be clockwise or counterclockwise. So if you are moving this direction, this is clockwise. This direction, it can be anti-clockwise, suppose. So in this, you will find histidine, threonine, STR. Actually, this is the same sequence. So you will again draw the same line. It is actually in this. It is moving from the donor into the recipient in this direction. So if you write STR, after STR, you are having ILV and threonine. So you can have this particular. So what will you do? You will again write ILV here, and then you will add threonine, right? So ILV, then you will write threonine, THR. Then what will you do? You need to see this also. So threonine, and then again it is showing this particular sequence, and there is nothing, I think. Okay, so threonine, here also you have threonine. Okay, so THY, histidine, tryptophan you already have covered, and LIP and threonine, right? So you need not to go into the details of fourth strain. By this only you will able to arrange them. So this is threonine, LIP, tryptophan, histidine, threonine, STR. STR is from this particle. This is the last letter of this. So STR, and you will choose these three. Okay. So this first strain and three letters from the third strain. Now, this is the linear sequence. So you will find it here. Threonine, LIP, TRP, tryptophan, histidine, THY is threonine, I think. No, sorry, this is not threonine. This is something else because threonine is this one. Last three letters of this. So this is having STR, ILV, and because it is having again threonine, so you can think of that this is a circular chromosome. Circular. So you can link this in this fashion, right? So once you find this linear fashion in which this will be transferred. So this is the order of transfer from donor cell to some recipient cell, right? There is second problem. Sorry, I need to remove these lines first. So second problem is also the same kind, but here instead of writing them three-letter codons, here they have written ABCD. So what are these? These are HFRs. As I showed you one diagram where you are having three different HFR strains and you need to draw them like this. It is the similar problem where you are having actually 6 HFR strains, right? So if you are having 6 strains of HFR, so you are having PAQB, what you need to do each time, you need to match the letters. And what you need to make overall arrangement, out of this overall arrangement, you need to trace the order, right? So first of all, we will see the first one, PAQB. If you see the second, it is not having. Okay, so leave it. Now see the third one, BQ. If you see this and this and this, then it is having some commonality. For example, this is having PAQB, it is having QB. So QB, you can suppose F will come first and then E. So you will write F and E here. Guys, please ask me if you do not understand this because this is easy and essential too. So what I did, I'm making one linear arrangement, or actually I'm trying to trace in which order actually the genes can transfer from one HFR strain to some F negative strain, right? So it is showing one first HFR is PAQB, third HFR is EFQB. So if you see this, it is showing the QB, and you will trace QB should be followed by some third letter, that is F here. So I write F here, and then E, I write E here. Then the second will give you some information, second strain or the second HFR. It is showing FEZC. Actually, this is anti-clockwise, so I will write Z and C there. So ZC, right? Then I will see this one and I will try to match my sequence. It is showing it is very similar, and it is showing that CZ or FEZC. It is showing this one only. So we can skip this. Then we have fifth HFR. So this is showing ZC and WD. So you can think of W will come here followed by D, right? Then again you have AP. So you will move in this direction now. AP DW. So it will give you the one common sense to use. So AP DW. So it will become circular. Mostly the bacterial chromosome are circular, therefore you will get the same order again. So this will be your gene sequence, gene order. So the different HFR strains which are given to you, we will try to trace the sequence either clockwise or anti-clockwise in whatever direction they are given. We will try to put them in a single arrangement or linear arrangement, and then we will try to match them with the options. If we are having an proper option which are matching with our sequence, that will be our final option. So what we have decided, PAQB followed by FEZCWD. So actually I cannot, okay, so as you will see here, this is showing AP. So you can trace it from here, AP, and this is followed by DW, CZEFQB, BQ, sorry, BQ. So actually, if you read this in this particular order, you can write your sequence, this particular sequence as, uh, okay, so let me write this. So it is from here you can write, because this is the option AP, then DW, CZ, EF, DQ. This will match your one of the options, and actually this is the order. So we will find the real answer, right? Now we are having one more question, right? So we are moving to MCQ, not one. Actually, we are having more than one questions there. Are you able to understand this? This is these type of questions are very easy to do actually. So the second one is here. Suppose this is a question. Okay, so the four HFR strains of actually, this has been asked in GATE. So, four HFR strains of E. coli were generated from the same. Whatever the question is written, you need not uh, read. Even you can skip the question. Only you need to focus on these strains which are given to you. These are HFR strains, and then you need to draw your linear pattern out of it. So, I'm having the line. So, okay. So what I will do, DQ, I will move in this direction. So I will have this, right? So DQWMT. And if you have this M here, T here already, you can suppose the P will come X and then A, PXA. So you will write PXA here then. So I'm writing, sorry, PXA, which is coming from here in the from the second string. So the PX and then A. In this order we are moving. Then the third one is BNCX. So we are having X here, A here. Then it will show you that C after A, C will come, N and then B. So I will write again, sorry, C and B, right? And then in the last one, BDQ, so you can think that BD, and then it is following the same order. It will give you this glimpse that, oh, now it is circularized now. So BDQWM. So now you are having one single bacterial chromosome in which you know the order of genes, and now you will try to match them. So DQWMT, EXAC, and B, this is showing the correct order. If you try to find some other, for example, AX, so you will having this AXP. So first of all, you need to remember it can be clockwise or counterclockwise. So in both the directions you need to match, but actually the order will not match at all. So AXP, TM, DQ, it is having DQ, but this D is after Q actually, but it is showing D first. So this cannot be the case. Similarly, these two. Okay, so the correct option is A, which is matching with our this particular. So that's how, if you are having different HFR strains, you need to find a linear pattern out of it, and then you will match with the options. If you are matching it correctly, that particular option will be your correct option. Okay, so because we have covered all the three processes, transformation, transduction, and conjugation, so we are having some questions from transformation also. For example, right, the first will be the option because it is showing the correct linear pattern, right? The 16 is transformation in S. pneumoniae or Streptococcus pneumoniae involves the binding with the double-stranded DNA fragment, is a random process which is virus mediated. It is a natural process, involves the cleavage of one of the strand of the DNA by exonuclease. So, because most of you have attended the all the three gene transfer classes: transformation, conjugation, and transduction, right? So, what will be the correct option out of that? And she's saying it is B. Okay. So R, it is a natural process, right? S, it is degrading one of the strands. Okay, right. What is the problem with B? So actually, this is the C. Okay. So actually, all the options are correct, but not the B, because it is not a virus mediated process. If it was a virus mediated process, it's called it is not called the transformation. This was called transduction, right? So this is not correct. Otherwise, all are okay. I'm having one more question for you, for example. Okay, so I'm having this. This is very interesting. So 12th, bacterial conjugation can be used to map the genes on bacterial chromosome. We can use, as we have studied recently, which of the following would be the best donor strains? And actually, this has been asked in IIC. Are you not able to get an option out of it? I just told you. Okay, so we have discussed this particular question recently. Only we have, so we were discussing this. For example, if it is having all the genes, for example, A plus, B plus, C plus, ABC can be anything. And for donor, it should be sensitive, right? Otherwise, it will not be selected. You cannot select the recipient, right? You will select recipient on the basis of these resistance. So if it is sensitive, then only it can be a donor. So now you can think of, it should be sensitive first of all, so it can be either A or B. But since it is having all negative and it is having positive, so B should be the answer, because it is having all the donor characteristics. It is sensitive, it is having all the ABC genes, right? So it can transfer all the three genes to the some recipient bacterial cell. Similarly, we are having other questions. You can go through them and try to find them. They are easy. Okay, so the last, what we are going to discuss in this particular class, we are going to, so it is given during conjugation, what will happen? So it will require cell to cell contact actually. So this is the option A. Which of the following processes would not contribute? So, okay, so whatever question you have in examination, you need to underline these words if you are having not, if, any, or. These words are very important. They can change the meaning of any question, right? If you skip this not, you can uh, answer a wrong option, right? So this is asking not contribute to the genetic variation within the bacterial population, because we know all the transduction, transformation, conjugation, mutation. These processes are involving two, transduction and transformation. These are gene transfer processes. So they will responsible for, they will contribute for genetic variations. Mutations also, they will responsible for genetic variations. But actually this meiosis is not reported in bacteria. There is no gamete formations, and therefore this will be the correct answer, right? D. So as I discussed with you today, we will discuss M13 also. I will tell you the important features of lambda phase as well as M13. Both are very important in cloning experiments, right? So first of all, we will discuss lambda phase. Lambda phase is a temperate phase. What is meaning of temperate phase is it will undergo lysogenic cycle, and then at some stage of life cycle of bacteria, it will show the lytic cycle, right? So in the lysogenic cycle, it will integrate its chromosome into the bacterial DNA, bacterial chromosome. The lambda genome, since lambda, what is lambda? Lambda is this phage, is this virus is called lambda. As I showed you in the beginning of the class also, so there are two bacteriophages discussed in this book. One is lambda phase, and this is lambda actually. This is M13, which is a filamentous virus. These viruses are called bacteriophages if they infect bacteria only. So what will the basic features of these lambda phages? These are having double-stranded DNA, but this DNA can be present in this linear form as well as in circular form at some stages of their life cycle. So generally in the bacteriophage, in the virus, this is present in the linear form, and once it will come into the bacterial cytoplasm, it will be one possibility is it will be replicated, and the second possibility is it will become circularized so that uh, it can be integrated into the bacterial chromosome. So it is integrated into the, so it is injected into the bacterial cell. Now it will become circular so that it can be integrated into the bacterial chromosome to make prophase, and then with the cell divisions it will move to the daughter cells also, right? At some stage of cell, then it will come out of the bacterial chromosome, and then it will do the lysis of the bacterial cell. Now you need to understand that E. coli cell, which is the host of lambda phage, it is having 20-minute life cycle, and therefore you can assume that the life cycle of virus which is infecting the E. coli should be less than 20 minutes. So within 20 minutes, it need to complete its life cycle. Second important thing is what is so important that this is present in linear as well as in circular form. So there is a 12 nucleotide stretch that is called complementary sites or the cohesive ends, from where this double-stranded DNA is single-stranded, as I will show you. So suppose this is the lambda chromosome. It will replicate by rolling circle mechanism. And what is the importance of these cos sites? One thing is they will be required uh, for the circularization of the linear chromosome, right? So here it is circularized, but since it is because of these cos sites, they are complementary 12 nucleotide base pairs, which are CG rich or the GC rich sequences, which will help in circularization of the linear chromosome. And this circularization is important for the integration within the bacterial chromosome. Second important point you need to remember is these cos sites are the points where actually when this replication is happening, so this is the lambda phage genome, which is 49 kb in size. So when this will replicate by rolling circle mechanism, it will not, so it will continue to replicate only when several copies are formed, or these are called catenae. Catenary is a chain in Latin, it meaning chain. When a chain of these bacterial chromosomes, of these lambda fast chromosome is being formed, then these cos site will act as recognition sequences for some endonucleases. For example, endonuclease A. So as it is written here, gene A endonuclease. So endonuclease will cleave these cos sites and they will separate these viral chromosome or viral DNA. Then only these cohesive ends will come together. They will make a circle like this. So generally in the bacterial cell, they are present in this circular form, from which make the complementary base pairing between these cohesive ends or cos sites. So now you are familiar with the two functions. One is these cos sites are required for the circularization of chromosome. But before that, when the replication is actually happening, these cos sites will act as recognition sequence for endonuclease A, which are joined together by these cos sites. So generally the chains of the viral genome are formed continuously in rolling circle mechanism of replication. Now, the second important bacteriophage which can be used to clone the genes or to transfer the genes from one bacteria to some other bacteria is M13. M13 is a filamentous phage, as we have seen the structure. Uh, so it is the M13 phage. Now, one important point I already told you that the infection process is through the pilus. So it will use some surface appendages of E. coli through which it will inject its DNA into the bacterial cell. And the second thing you need to remember is this is a single stranded DNA. In the lambda phase, we were discussing double stranded DNA, but here this M13 is having single stranded DNA, which will become double stranded replicative form. So double stranded DNA, which is formed within the bacterial cell, is then called replicative form. And then it will undergo replication, right? So, and second thing is M13 DNA is not integrated into the bacterial chromosome, unlike lambda phase chromosome. Third thing is it will continue to secrete the bacteriophages or viruses from the E. coli cell. So it will secrete them out continuously. And what should be the number? Generally, these replicative form or these chromosome or these genome are present up to 100 copies per bacterial cell. So in lifetime, in the 20-minute lifetime of E. coli, they can secrete up to 1,000 copies, up to 1,000 copies of M13 virus. Okay, so because they can secrete the virus, they need not lyse the bacterial cell. First of all, they will not integrate into the bacterial chromosome. Second thing is they do not lyse the bacterial cell. They will continue to secrete the viruses out of this E. coli cell. Now, the injection of an M13 DNA molecule into the E. coli cell occur via the pilus, the structure that connect the two cells during the sexual conjugation process. We have studied this. Phage particles are continuously assembled and released, about 1,000 new phage particles being produced during each generation of an infected cell. Now, what are the benefit of using this M13? M13 is being used as a cloning vector because it is having a small size of its genome. It is made up of only 6,407 nucleotides. So this is 6.4 kb. And if you know the characteristics of an ideal cloning vector, it should be less than 10 kb. So actually this is having small size. Small size is having one disadvantage. It can accommodate only a few genes. It is having small room to accommodate a few genes only. But the advantage is since it is having small size, so it can be used as a cloning vector, right? Whereas the large DNA is having some purification problems and it is hard to manipulate them. Now, third important thing means the last important thing of this M13, using of M13, because the single stranded DNA is being transferred to the bacterial cell and then only it will make the double stranded DNA replicative form. So this single-stranded DNA can be used to achieve the single-stranded DNA copy versions of different genes. So if you want to clone some gene, if you want to purify, or if you want to remove the single-stranded version of that particular gene, you can clone that particular gene into the M30 bacteriophage. It will give you the single-stranded version of that particular gene. So this is one advantage of using this M30, right? What is the another advantages why we need to use this? So because several features of F13 make this phage an attractive model for cloning vector. The gene is having less than 10 kb in size, only 6.4 kb, within the range desirable for the potential vectors. In addition, double-stranded replicative form of M13 genome behaves very much like a plasmid. So when it will make a double-stranded version of its genome, it is very similar to the plasmid, which is present in the bacterial cell and can be treated as such for experimental purposes. It is easily prepared from the culture of infected E. coli cells. So you can separate the plasmid from the bacterial culture. Similarly, you can separate these replicative forms from the E. coli cells, and then you can use these replicative forms to do some kind of experiments. What is the basic use of these double-stranded versions of these replicative forms of M13? Single-stranded versions of cloned genes are obtained. So the most importantly, genes cloned with the M13 based vector can be obtained in the form of single-stranded DNA, right? This I told you. These single-stranded versions of cloned genes are useful for several techniques. One is DNA sequencing. You can perform the DNA sequencing using these single-stranded genes. Then in vitro mutagenesis, you can create mutagenesis. You can create mutations in these genes, and then again you can infect these bacteriophages, some E. coli cells with these bacteriophages. So M13 vectors are also used in phage display. What is this? This is a technique in which you identify pair of genes whose gene products, for example, protein products, interact with one another. So in phage display, in DNA sequencing, in vitro mutagenesis, so these are having various different applications. So can be used to get the single-stranded versions of DNA versions of some of the genes, which can be used for gene sequencing and other applications. For example, phage display to know the gene interactions and to know the and to perform the mutations or mutagenesis experiments, right? And then some of other cloning vectors which are being used in various species. For example, adenovirus are used in humans, baculovirus are used in insect cells, and in plants we generally use colimoviruses and Gemini viruses. So these questions has been asked in various competitive examinations. So you need to remember adenovirus human cells gene therapy experiments में use करते हैं, baculovirus insect cells में अगर pharmaceutical protein production और इन सब में use होता है. Polymovirus and Gemini viruses are being used in cloning, which are very specific for plants, right? So with this, we are ending today's class. I hope you are able to understand all the three ways of gene transfer: uh, conjugation, transduction, and transformation. So I have given much time to discuss this particular topic because this is really important and question has been asked uh, this from this particular section. In next class, we will start some new topic as we need to finish the microbiology in our upcoming classes. Thank you. If you are having some query, please ask me. Priyanshu is having no queries. Good. Priyanshu, you are not asking me any queries, means you are not solving MCQ and Catalyst and other books which are being shared in the group. Sir have already shared these books in or online group in WhatsApp group. So you need to solve these books at least. At least. Okay, no problem at all. Thank you so much for the class. And if you have queries when you solve these questions, please feel free to message me or WhatsApp or whatever you want to. Okay, thank you. Good night.